Hello everyone, welcome back. Today I'm here with this topic of cell biology, that is cell wall. In this presentation I will discuss about the composition of cell wall, types of cell wall, and what are the various functions which are performed by a cell wall. So these are the topics that I will try to discuss in this presentation. Let's dive in. Firstly, cell wall. As we know that in certain groups of organisms, like we have protists, fungi, plants, and bacteria that have cell wall, and this cell wall helps in maintaining the definite shape, and it's also a protecting structure that protects the organism from various stresses. So it provides definite shape, rigidity, strength and protection against mechanical stresses. So बहुत इंपॉर्टेंट रोल प्ले करता है सेल वॉल, जिससे डेफिनेट शेप होती है सेल की. इट प्रोवाइड मैकेनिकल सपोर्ट and it acts as a protecting structure from mechanical stresses. So it is present in the cells of plants, fungi, protists, and most of the bacteria. So it is found in all these organisms. There are some exceptions, like we have animal cells and heterotrophic protists. They do not contain cell wall. जो animal cells हैं और heterotrophic protists हैं, इनमें cell wall missing होती है. They don't have cell wall. So cell wall of prokaryotes, fungi and algae and plants are distinct from each other in their microscopic structure and chemical composition. तो ये जो सभी organisms हैं, इनकी cell wall का जो chemical constitution है and their microscopic structure, they varies from organisms to organisms. As we know that जो plants होते हैं, उनकी जो cell wall है वो बनी होती है ग्लूकोज के एक पॉलीमर से, जिसे कहते हैं हम सेल्यूलोज, which is the most abundant organic matter on this earth surface. सबसे ज़्यादा पाया जाने वाला organic matter है ये earth surface पे. Then fungus की जो cell wall है, that is made up of chitin. Then we have bacteria, जो कि prokaryotic cells हैं. They have some special type of cell wall which is made up of sugar plus amino acid, that is peptidoglycan. So the plant cell wall is composed of primarily strong fibers of carbohydrate polymer. Carbohydrate polymer में है नाम cellulose, cellulose से बनी होती है. और जो बैक्टीरियल सेल वॉल है वो बनी होती है शुगर और अमाइनो एसिड से, जिसके पॉलीमर को कहा जाता है पेप्टाइडोग्लाइकेन. एंड फंगल सेल वॉल इज मेड ऑफ काइटिन, ग्लूकेन्स एंड प्रोटीन्स. सो जो केमिकल कंपोजिशन है इन डिफरेंट ग्रुप्स में सेल वॉल की, दैट इज डिफरेंट. नेक्स्ट कम स्ट्रक्चर. मेनली जो सेल वॉल है ये डिफ्रेंशिएट होती है तीन मेजर टाइप्स में. वी हैव प्राइमरी सेल वॉल, देन सेकेंडरी सेल वॉल और मिडल लैमेला. तो ये जो स्ट्रक्चर है विच इज़ प्रेजेंट बिटवीन द प्लाज्मा मेम्ब्रेन एंड मिडल लैमेला, इसे कहा जाता है प्राइमरी सेल वॉल. इन सम ऑफ द टिश्यूज़, मेनली जो मैकेनिकल टिश्यूज़ प्रेजेंट होते हैं प्लांट्स में, जैसे कि वुड है, वुड में जाइलम ट्रैकीड्स होते हैं, उनमें कुछ अंदर की तरफ को जो प्राइमरी वॉल है इसके अंदर की तरफ को और डिपोजिशन होती है केमिकल्स की. दे फॉर्म द सेकेंडरी सेल वॉल which mainly contains carbohydrates, means sugars, and that is cellulose plus hemicellulose. It also contains xylanes and lignin. So firstly I will discuss about the primary cell wall. जो primary cell wall होती है ये लाई करती है between middle lamella and plasma membrane. जो plasma membrane है और middle lamella के बीच में जो wall present होती है, that is called primary cell wall. So primary cell wall forms initially during cell growth. It is located between middle lamella and plasma membrane. और दूसरी जो इसकी स्पेशल चीज़ है, ये प्रेजेंट होता है इन यंग ग्रोइंग टिश्यूज़ और ऐसे सेल्स लाइक वी हैव पैरेन्काइमेटस सेल्स या मेरिस्टेमेटिक सेल्स जो कि थिन वॉल्ड होते हैं और कॉन्टिन्यूसली उन्होंने ग्रो करना है, तो ऐसे सेल्स में प्राइमरी सेल वॉल प्रेजेंट होती है. सो दिस इज़ थिन, इट इज़ परमेबल एंड इट इज़ इलास्टिक एंड एक्सटेंसिबल, एडिंग सेल वॉल्यूम इंक्रीज, जो कि हेल्प करता है, इलास्टिसिटी हेल्प करती है सेल की ग्रोथ के लिए, जैसे सेल साइज में इंक्रीज होते हैं. एंड इट इज़ फाउंड इन द पैरेन्काइमेटस सेल्स एंड मेरिस्टेमेटिक सेल्स, एज वेल एज वी हैव सम स्पेशल टाइप ऑफ सेल्स लाइक लीफ सेल्स. फ्रूट्स में, कॉर्टेक्स में और पिथ सेल्स में प्राइमरी सेल वॉल प्रेजेंट होती है. और जो ये इसकी केमिकल कंपोजिशन है, इट कंटेन्स 25% सेल्यूलोज, 25% हेमीसेल्यूलोज, 35% pectins and 1 to 8% some special structural proteins. तो ये इसकी chemical composition है. If we talk about structure, it consists of cellulose microfibrils in a gel-like matrix of hemicellulose fibers. मीन जो मैट्रिक्स है वो बनी होती है हेमीसेल्यूलोज से, और जो माइक्रोफाइबर्स हैं सेल्यूलोज की हैं, ये लाई करती हैं इसमें. and it also contains pectin, which act as a filler material. Then it contains some glycoproteins. So हेमीसेलुलोज लिंक्स माइक्रोफाइब्रिल्स टू द मैट्रिक्स. हेमीसेलुलोज का काम होता है माइक्रोफाइब्रिल ऑफ सेलुलोज को लिंक करना मैट्रिक्स के साथ, जबकि जो पैक्टिन है वो एक्ट करता है एज अ मैट्रिक्स फिलर. तो मैट्रिक्स को फिल करने का जो काम है वो पैक्टिन करता है. देन जो ग्लाइकोप्रोटीन्स होती है दे एक्ट एज गाइड फॉर माइक्रोफाइब्रिल ओरिएंटेशन. जो ये माइक्रोफाइब्रिल्स होती हैं जो कि सेलुलोज से बनती हैं, उनकी ओरिएंटेशन किस तरह की होगी सेल वॉल में, उसमें हेल्प करती हैं ग्लाइकोप्रोटीन्स. थिनर रीजन्स इन द प्राइमरी सेल वॉल आर प्राइमरी पिट्स. जो प्राइमरी सेल वॉल है उसमें कुछ रीजन्स होते हैं जहाँ पर डिपोजिशन कम होती है. दे फॉर्म थिन स्ट्रक्चर, मीन्स दे फॉर्म सम स्ट्रक्चर्स जिन्हें कहते हैं प्राइमरी पिट फील्ड्स. और यहाँ से जो एक साइटोप्लाज्मिक कनेक्शन होता है दूसरे सेल के साथ वो निकलता है, दैट इज कॉल्ड प्लाज्मोडेस्माटा. सो साइटोप्लाज्मिक कनेक्शंस थ्रू दिस पिट्स कॉल्ड प्लाज्मोडेस्माटा. इन्हीं पिट से ये जो साइटोप्लाज्मिक कनेक्शंस हैं वो बनते हैं जिन्हें कहते हैं प्लाज्मोडेस्माटा. ये हेल्प करते हैं कनेक्टिंग द सेल्स इन मल्टीसेलुलर प्लांट्स, और इसमें वेरियस सब्सटेंसेस का एक्सचेंज होता है थ्रू दीज स्ट्रक्चर्स, बेसिकली सिम्प्लास्ट मूवमेंट. देन सेलुलोज जो होता है दैट इज़ अ पॉलीसैकेराइड. दिस इज़ अ होमोपॉलीसैकेराइड. ये एक होमोपॉलीसैकेराइड है. ये बना होता है अल्फ़ा डी ग्लूकोज मॉलिक्यूल से, जिसमें लिंकेज होती है दो ग्लूकोज के बीच में, एंड दिस लिंकेज इज़ नॉन एज अल्फ़ा 1,4 ग्लाइकोसिडिक लिंकेज. ये बनाते हैं वी हैव, अगर 100 चेन्स हैं सेलुलोज की, 100 चेन्स ऑफ सेलुलोज फॉर्म्स वन मिसल. ये बनाता है एक मिसल, और 20 मिसल्स बनाते हैं मिलके एक माइक्रोफाइब्रिल, और 250 माइक्रोफाइब्रिल्स बनाते हैं एक बड़ा फाइबर जिसे बोलते हैं मैक्रोफाइब्रिल या सिर्फ फाइबर. सो इन दिस वे जो सेलुलोज है उसकी फर्दर पैकेजिंग होती है, एंड दे हेल्प्स इन फॉर्मेशन ऑफ दिस प्राइमरी सेल वॉल. नेक्स्ट वी हैव सेकेंडरी सेल वॉल. सो इट इज़ फाउंड इन सम मैकेनिकल टिश्यूज़, जैसे कि वुड के साथ एसोसिएटेड स्ट्रक्चर. वी हैव ट्रैकीड्स, उनमें ये प्रेजेंट होता है. तो सेल वॉल जो कि बनती है जब प्लांट के सेल ने ग्रोथ पूरी कर ली होती है. सो सेल वॉल दैट फॉर्म अ वॉल आफ्टर सेल ग्रोथ और वॉल एक्सपेंशन हैज़ बीन सीज्ड. जब प्लांट का सेल पूरी तरह से ग्रो कर गया है और उसकी ग्रोथ रुक गई है, तब सेकेंड वॉल बनती है, जिसे कहते हैं हम सेकेंडरी सेल वॉल. सो दीज वॉल्स आर थिकर, more rigid, and provide more protection to the plant. So they are mainly meant for mechanical support, and it exists between the primary cell wall and the plasma membrane. ये पाई जाती है जो primary cell wall होती है, this is primary cell wall, और we have this inner to this we have plasma membrane. So plasma membrane और primary cell wall के बीच में जो structure present होता है, this is called secondary cell wall. इसकी different layers हो सकती हैं. There may be 3 or more layers, जैसे S1 है, S2 है, S3 है, SX. तो ये जो layers हैं ये हो सकती हैं secondary cell wall में, और ये lie करता है in between primary cell wall and plasma membrane. So this wall is laid inner to primary wall by deposition of materials over the surface of existing structures. The xylem cells found in wood are more prominent, having extremely thickened secondary walls, and they are strengthened by one chemical substance that is called lignin. तो जो सेकेंडरी सेल वॉल्स हैं उसमें लिग्निन भी होता है, दैट इज़ पॉलीमर ऑफ एरोमेटिक अल्कोहल. ये एरोमेटिक अल्कोहल का पॉलीमर है. इट इज़ मेड अप ऑफ एट लीस्ट 3 लेयर्स ऑफ और सम टाइम मोर. तो जो सेकेंडरी सेल वॉल होती है ये बनी होती है 3 लेयर्स से, जैसे कि हम यहाँ पे देख रहे हैं, जैसे S1 है, S2 है, S3 है. इसके अलावा कई बार इसमें ज़्यादा लेयर्स भी हो सकती हैं. सो दे आर नेम्ड एज S1, S2, S3, SX, etc. Secondary cell walls, they are thick, and they are found in cell like we have tracheids, fibers. Here the cell wall is multi-layer. So जो wood है that is secondary xylem. So its secondary wall contains xylenes, more cellulose, and neatly aligned microfibrils of cellulose. It also contains lignin, which is a polymer of aromatic alcohol in secondary walls, और ये provide करता है extra support to the plant, and also imparts the defense to form the defense system of the plant cell against insects or pathogen. तो ये strength provide करता है lignin secondary cell wall को. Then cellulose microfibrils orientation varies in secondary wall layers. जो different layers है उसमें cellulose के microfibril की orientation vary करती है. Sometimes there may be a third layer, that is called tertiary cell wall, which is beneath the secondary cell wall and has distinct morphology, chemistry and staining properties. तो इसमें ये जो tertiary cell wall है ये different है secondary cell wall से in its morphology, chemistry और staining properties, और ये बनी होती है cellulose और xylene की. So that is called tertiary cell wall. So firstly we have this primary cell wall which lies between middle lamella and plasma membrane. Then we have the secondary cell wall which lies between primary cell wall and plasma membrane. And sometime there is another layer which is present between secondary cell wall and plasma membrane. So this is called tertiary cell wall. और ये जो tertiary cell wall होती है ये vary करती है in its chemistry, morphology and orientation of or staining properties of this layer. So lignin एक most abundant aromatic biopolymer है अर्थ में, और ये इसका जो important role है, it helps in protecting the plant from insects or pathogenic. Again, this is the diagram which shows this primary cell wall which lies between middle lamella and plasma membrane. Then we have the secondary cell wall which lies between primary cell wall and plasma membrane, and it is mainly composed of various chemical substances like we have cellulose, hemicellulose, lignin, pectin and structural proteins. Next comes important structure, that is middle lamella. So this is a cementing layer which is present between the two primary cell wall. जो दो primary cell wall हैं उनके बीच में जो ये cementing material है इसी को कहा जाता है middle lamella. So thin layer of material at the junction where the walls of adjacent comes in contact with each other. So it is present between two primary walls of adjacent cells, और जो इसकी composition है that varies from the rest of the cell wall, and it contains high concentration of pectin. इसमें होता है ज़्यादा pectin, और ये बना होता है pectate of calcium and magnesium से. Pectic acid is large polygalactouronic acid, means polygalactouronic acid है pectin, और इसका जो monomer है ये बना होता है alpha D galactouronic acid से. So it is made up of alpha, this one, that is alpha D galactouronic acid, और इसके बीच में भी होती है linkage, that is alpha 1,4 glycosidic linkage. So it acts as a cementing material. ये एक तरह से cementing material act करता है और दोनों cells को attachment में help करता है. So it also forms a boundary between the cells. So this is the pectin which forms the cementing layer that is called middle lamella, और इसका जो monomer है that is called galactouronic acid. Next comes plasmodesmata. So desmos word का मतलब होता है ribbon और ligament, और ये connect करता है neighboring cells को. So इसका जो primary objective है, this is a cytoplasmic connection between two cells, और ये बनता है जहाँ पे primary cell wall की deposition कम होती है जिन्हें बोलते हैं pit fields. So they form a protoplasmic band called symplast, and it is generally cylindrical membrane lined channel with a diameter of 20 to 40 nanometers, and it consists of a canal lined by plasma membrane which remains continuous with elements of smooth endoplasmic reticulum, membranes of each connected cells. Plasmodesmata features a simple or branched tubules जिन्हें कहते हैं desmotubules, और desmotubule एक extension है endoplasmic reticulum की, and the connecting cells smooth endoplasmic reticulum elements, and it facilitates cell communication. Main function is cell communication by allowing passive transport of small molecules and active transport of proteins and nucleic acids between adjacent cells. They are essential for stimuli communication, and there are two pathways for plasmodesmata biogenesis or formation of plasmodesmata. First one is the primary one form during cell division, originating from phlegmoplast से बनता है. They are basically microtubules, उनके arrangement से ही बनता है ये. Then while secondary one that dwells independently in pre-existing cell walls. दो तरह से ये बनते हैं. The next comes pits. Pits are small perforations which are meant for exchange of material, और ये बनती है primary cell wall पे जहाँ पे secondary wall deposit नहीं होती है. So areas on the cell wall where a secondary wall is not deposited, so those portions are known as pits, and each pit consists of a pit chamber and pit membrane. दो चीज़ें होती हैं, वन इज पिट मेम्ब्रेन एंड अदर इज पिट चैम्बर। तो दो चीज़ों से मिलकर बनता है एक पिट। एंड जो पिट मेम्ब्रेन है दैट कंसिस्ट ऑफ टू स्ट्रक्चर्स, दैट इज मिडल लैमेला एंड प्राइमरी सेल वॉल। तो दो चीज़ों से मिलकर बनता है पिट मेम्ब्रेन। So pits assist in translocation of substances between two adjacent cells, and commonly pits develops in pairs. And pits are of two types. One is simple pits, another is border pits. Firstly comes simple pits. So the pit cavities which are uniformly wide, जिनका अंदर से ल्यूमन एक ही डायमेंशन का होता है, on the side in the simple pits, and the pit cavity is not covered by secondary cell wall. और इसकी जो pit cavity है, पिट अपर्चर है, ये कवर नहीं होता है बाहर से सेकेंडरी सेल वॉल्स। सो इस तरह के पिट्स को कहा जाता है सिंपल पिट्स। व्हाइल इन केस ऑफ बॉर्डर पिट्स द सेकेंडरी वॉल बेंड्स लाइक आर्च ओवर द पिट एंड प्रोवाइड्स अ बॉर्डर लाइक अपीयरेंस, जिससे ये दिखता है बाहर से बॉर्डर की तरह। एंड दिस बॉर्डर लाइक अपीयरेंस इज नोन एज, that gives its name, that pits are known as border pits. And the portion of middle lamella crossing the pits becomes thickened. जो middle lamella का portion होता है ये thick हो जाता है, and that forms a central structure which helps in controlling the flow of material. So this central structure, this is known as torus. इसे कहा जाता है torus. And this thread-like structure which holds this torus, इसे कहा जाता है margo. That structure is known as margo. Next comes the ultrastructure of cell wall. So as we know that it consists of various biomolecules, like we have cellulose, hemicellulose, pectins, compounds and lignins. The cell wall is made up of microfibrils. Microfibrils बनी होती है cellulose से, और जो microfibril molecules होते हैं they are homogeneous in chemical composition, arranged in specific manner, and they are made up of cellulose molecule that I have already discussed. Structure units जो होती हैं cell wall की जिन्हें बोलते हैं microfibrils, और ये होती हैं flat ribbon like structures जो रन करती है पैरेलल टू ईच अदर, एंड their dimensions are they are 10 nanometer long and 3 nanometer thick and composed of near about 8,000 to 15,000 glucose monomers. और हम कह सकते हैं कि इसमें होती हैं 2000 glucan chains, and these chains of cellulose molecules are arranged in parallel to at some regions of microfibers and form a three-dimensional crystalline lattice जिसे बोलते हैं micelles. So near about 100 cellulose chains form the structure that is called micelle. So each cellulose microfibril consists of 3 micelles or elementary fibrils. Each micelle has about 100 cellulose molecules, and each molecule is made up of about 40 to 70 glucan chains. As we know that one microfibril is equal to 3 into 100 into 70, that is near about 21,000 glucan chains. Cellulose is a long chain, straight chain, that is a polysaccharide basically, और इसमें basic unit होता है D-glucose, and it is joined to another glucose molecule by this bonding that is called alpha-1,4 glycosidic linkage. An association of about 100 cellulose chains, 100 cellulose की chains एक structure बनाती है जिसे बोलते हैं हम micelle, and 20 micelles they constitute to form microfibril, while 250 microfibrils forms this larger structure that is called fibril. So here we can understand basically 100 chains of cellulose form the structure that is called micelle, and the micelles aggregate. There are 20 micelles that aggregate to form microfibril, and 250 microfibrils they will form this larger subunit that is called macrofibril or fibril. Hemicellulose binds with microfibrils, binds with microfibril, and it binds it with matrix. And pectin is the filler substance that I have already discussed, and numerous microfibrils are aggregated to form macrofibril. तो बहुत सारी जो microfibrils हैं वो combine करती हैं और बनाती है बड़ा fibril जिसे बोलते हैं macrofibril, और इसका dimension होता है near about 0.4 micrometers wide and composed of near about 5 lakh cellulose molecules. Numerous macrofibrils are aggregated to form the cell wall. Finally, they will form the cell wall. Next comes hemicellulose. So it is short but branched heterogeneous subgroup of polysaccharide. ये homopolysaccharide नहीं है, this is a heteropolysaccharide that are strongly bonded in the cell wall, and different tissues wherein there are hemicellulose composition. The most abundant hemicellulose, सबसे ज़्यादा पाया जाने वाला hemicellulose है primary cell wall में, that is present in dicots is xyloglucan. ये present होता है primary cell wall में xyloglucan, और specifically in dicots. और ये जो polysaccharide होता है, it has a backbone of alpha-1,4 glycosidic linkage of D-glucose per residues like cellulose. But xyloglucan has short side chains that contains two other sugars. One is xylose and other is galactose. So इसलिए ये जो sugar, ये जो biomolecules हैं polysaccharide, they are heterogeneous polysaccharides. These short side chains prevent the assembly of xyloglucans into crystalline microfiber. And secondary cell walls typically contain less xyloglucan. Secondary cell wall में कम होता है, but it contains secondary cell wall में ज़्यादा present होता है xylane and glucomanans. And they also binds tightly to the cellulose. So the cell wall of grasses, that is monocots, contain only small amounts of xyloglucans and pectins. इसमें कम होते हैं, ये ज़्यादा dicots में होता है, which are replaced by this another chemical substance, biochemical substance that is called glucouranoarabinoxylane. It consists of two types of bonding, that is 1,3 or 1,4 glycosidic linkage of the glucan. The next important component of cell wall is pectins. So it is made up of polygalactouronic acid, and it is the main component of middle lamella. So this is a heterogeneously branched polysaccharide that have numerous negatively charged D-galactouronic acid residues along with some D-glucouronic acid residues. तो दो तरह के सबयूनिट से बना होता है, that is galactouronic acid and D-glucouronic acid, and it is the main component of this is galactouronic acid and this is glucouronic acid. They combine to form this structure that is called pectin, and it is the major component of middle lamella, but also found in primary cell walls. It is a polymer of around 200 galactouronic acid molecules, and it is the most soluble polysaccharide and can be extracted with hot water. इसको hot water से extract किया जा सकता है. It normally contains acidic sugars such as we have galactouronic acid, and it also contains some neutral sugars such as ramenose, galactose, and arabinose. And this polysaccharides is also known as polygalactouronic acid that I have already discussed. Polygalactouronic acid is 1,4-glycosidic linked polymer of alpha D-galactouronic acid residues. When calcium ion is added to the solution of pectin molecule, it cross-links with gel to produce a semi-rigid gel and forms a cementing layer between two cell walls. Next important component includes structural proteins. So these proteins are divided into different categories on the basis of predominant amino acid. For example, we have hydroxychloroquine-rich glycoproteins, abbreviated HRGP. Then we have glycine-rich proteins, that is GRP, then proline-rich proteins, that is PRP, and arabinogalactane proteins, that is AGP. This hydroxyproline-rich glycoproteins are generally associated with cambium, phloem parenchyma, and various types of sclerenchyma. While glycine-rich proteins this proline-rich proteins are often localized to xylem vessels and fibers. Arabinogalactane proteins that forms near about less than 1% of diameter of the wall are water soluble proteins and very heavily glycosylated. इनमें बहुत ज़्यादा glycosylation हुई होती है, and they are present in very less amount. They are found associated with the plasma membrane and helps in cell adhesion and cell signaling during cell differentiation. So their main function is in cell adhesion and cell signaling. Next comes formation of cell wall. So cell wall originated de novo during the final stage of cell division when newly formed cell plates separates the two daughter cells. The cell plate formation takes place when Golgi vesicles and endoplasmic cistern aggregates in the spindle mid-zone area of dividing cell. This aggregation is organized by this structure that is called phragmoplast. So ये जो important structures and they are meant for this cell wall formation. Phragmoplast is a complex assemblage of microtubules, membranes and vesicles that forms during late anaphase and early telophase. तो ये जो phragmoplast है ये बनाता है microtubules से, membrane से और vesicle से, and they're formed during late anaphase or early telophase. The new middle lamella and primary wall are assembled from the contents of vesicles. जो नया middle lamella बनता है और primary cell wall बनती है, ये बनती है by the assembly of contents of the vesicle which are formed by this Golgi apparatus and endoplasmic reticulum. A newly formed wall can grow and mature through a process that is called, you can say, Firstly, it includes synthesis, then secretion, assembly, expansion in growing cell, cross-linking, and secondary wall formation. That leads to formation of this cell wall during after cytokinesis. The phragmoplasts disappear completely when the cell plate is completely formed. So these structures which are found here, they are the phragmoplast microtubules, and they help in formation of this septum or cell wall. Middle lamella is formed by deposition of calcium magnesium peptides. Primary and secondary walls are deposited just below the middle lamella. Depending upon the age of the cell, जो young cell होंगे उनमें primary cell wall होती है, and mature cells contain this secondary cell wall. और ये दोनों ही walls जो होती हैं, they are secreted by this protoplasm. So secondary as well as primary cell wall is secreted by protoplasm, and the secondary cell wall lies inner to the primary cell wall, while this primary cell wall lies inner to the middle lamella. The cell wall is very thin, delicate just after this formation and grow later by a deposition of the substances on the primary cell wall. There are two ways of the growth of cell wall. One is intussusception and another is apopposition. It is also known as accretion. Firstly comes intussusception. Here the cell wall material is deposited in the cell wall spaces. तो बीच में cell wall में कुछ जो spaces होते हैं, these spaces are filled by the deposition of cell wall material. This is called intussusception. It increases the surface area of the cell wall. Second comes apopposition. So it is the growth. It is the process by which the new cell wall material are deposited on the surface of existing cell wall. इसमें जो already cell wall है उसके surface पर एक और layer cell wall की material की deposit होती है. This process is known as apopposition. It is also known as accretion. It increases in thickness of the existing cell wall. So these depositions include, firstly we have lignin. This is the polymer of aromatic alcohol. It is glue-like substance that deposited in the cell wall of woody plants, and it helps in binding wood fibers, cells, and also provides strength. Water and dissolved material can pass through lignified cell wall, so it makes the cell wall permeable. Then deposition of lignin is known as lignification. This process को कहा था lignification. Next comes chitin, is a fat wax-like substance. So wax-like deposit which is found in the epidermal cells of the exposed surfaces. It makes the surface of the cell impermeable to water, and the deposition process is known as chitinization. Next we have suberin. So this it is a fat soluble deposit found in the secondary cell wall of cork cells. It makes the cell wall impermeable to water, and it also helps in checking the evaporation of water. तो ये water के entry का law नहीं करता है, and the process of deposition of suberin is known as suberization. This is the chemical structure of this suberin. Then next comes minerals. There are minerals like silica, which is deposited in the cell wall of grasses, that makes it hard, and they can cut the hands also. So calcium oxalate and calcium carbonates are found in the cell wall of a member of family Acanthaceae, Cucurbitaceae, and Moraceae. Mucilage, it is like carbohydrates that imbibe water and retain it for longer duration, mainly found in seed coats in the aquatic plants. So mucilage is covering that prevent the plant tissue from desiccation. Next comes some functions. So it mainly impart support to the plant. Cell wall provide mechanical strength and support to the plant. It also controls the direction of the cell growth. Then next is important function, that is resist turgor pressure. It provides resistance to turgor pressure and avoid breakage of cell. Turgor pressure helps the plant to remain rigid, erect, but too much of it can break the cell. Regulate growth and diffusion. The cell wall regulates growth functions of the cells, sending signals to enter the cell cycle and divide. The cell wall also assists diffusion. Being porous, allow the entry of some material and substances including proteins, so cell wall is highly permeable. Next comes protection. Again, this is a protective in function. It acts as a barrier for plant viruses and other pathogens. Essential for retaining water inside the cell. The next, it plays a very important role in cell communication. There are plasmodesmatas, which are cytoplasmic connection in the cell wall, which is serving as a connecting pathway among neighboring cells. Also allows transfer of products like we have nutrient ions and even waste materials to the adjacent cells. The next comes storage. Cell wall stores carbohydrates used in plant growth, especially in seeds. there are many questions related to this presentation. So this was all about for our today discussion about cell wall. I hope you will get some idea from this presentation. If you have any questions, queries, and any suggestions, give it in comment section. Thanks for watching. Have a nice time.